Hey everybody, we're going to go ahead and get started with the next part of the session, which features 3 talks, starting with Peter Winter, who comes to us from the Broad Institute. He is a PI there. So thank you so much for joining us.
Yeah. Well, thanks very much to the organizers. These are my personal favorite types of meetings because this is something that I wrestle with in our group that I'll kind of show in a minute. How do you actually get biologists to talk to mathematicians? You know, the idea of model being a word that means completely different things to both of those subsets of people is something we think about a lot. So today I'm also trying to lean into the prompt and kind of just pose like something that bothers me. And I would love to talk with you guys all about it. I'll show a little bit of data and kind of how we think about this problem, but it's largely meant for us to shout at each other. So I welcome that. Well, I think as probably everyone in this room recognizes, this is a really fun time to be masquerading as a biologist who kind of understands, well, masquerading as a computer scientist who is a biologist at heart. Anyways, like we're making these very large data sets at this point, right? And there's this convergence happening between what we measure and how we interpret it, right? And I think this is really apparent in this convergence of using modern AI and single cell genomics, right? So I grew up in a wet lab environment, learning how to just do Western blots and site-directed mutagenesis. Went on to MIT to learn computational biology, and now I kind of pretend to be able to do both. And so this is a really interesting time, at least for me as an early investigator, to attempt to try and stitch these two together. And so I think, as again, everyone in here is pretty well aware, we've gotten really good at measuring a lot of different things at scale about single cells over the past several years. We can measure the multiome at this point. We can measure hundreds of thousands of cells from different tissue types. This gets to be a really complicated problem computationally and biologically. This is obviously an out-of-date slide at this point, right? 2,000 total cells from a biopsy is modest by many estimates, but this is upwards of 44 million data points to consider if we start to think about all the different parameters that are housed within that very modest data set. And we've gotten really good, again, at measuring these across all these different tissue types, right? This is a very, very limited snapshot of these single cell data sets that I saw as a postdoc when I was in Alex Shalick's lab. So what ends up being true now is that no longer can I just be a biologist or I can just be a computer scientist and really bring these two disciplines together. So this interpretation of these data sets now becomes equal parts biology and computer science. And so kind of in keeping with the spirit of this meeting, I want to start with a little bit of a story about how my group actually was incepted. My buddy Lauren Crawford and I were in the same lab at Duke University together, where his hilarious setup, he's a statistician by training, his bench was in the wet lab, and it was right next to our Western blot station. He had this giant whiteboard that he would just always be writing on. And we would ask each other really stupid questions, like, he would walk up to me and say, well, why are you guys constantly dunking pieces of paper into milk when we were running our Western blots? And then I would ask him questions like, why do you guys constantly write only in the Greek alphabet? And so over the course of, we had 5 years to kind of figure out how to speak to each other, basically. But then what we've done since is now they've kind of put us in charge of helping other people figure out how to do this across our different disciplines. So our group is a collaboration between the Broad Institute and Microsoft Research, where my colleagues Ava Amini and Shri Raghavan have also joined us. Shri is a medical oncologist by training, and Ava is a machine learning expert as well. And so we've got a really tight-knit group of folks across the 3 different places where we all kind of meet at the Broad and do kind of similar things to what we're trying to all discuss here today. How do you actually get the biologists and the computationalists to talk to each other in effective ways? And so in the group, just kind of oriented to how we think about cancer cells, we largely work on cancer, although we have some dabblings in normal biology as well. I'm a cancer biologist by training. Shri's a medical oncologist, so this is kind of where that bend comes from. The way that we think about cancer cell phenotypes, though, is that they're much more complex and have diverse inputs than we've kind of been maybe traditionally trained. At least when I was first starting out learning cancer biology, oncogenes, tumor suppressors, right? It seemed very simple, but it's really not that simple, obviously. Cell state is the way we kind of think about these phenotypes now, and that is an integrative representation of many different cancer cell features. where obviously we've got the DNA sequence that's mutated. We have RNA expression, which is then the dynamic interpretation of that mutated genome, which leads to all the functional states, right? There's a lot of different inputs that are going to determine cell state here, DNA mutations being one of them, but epigenetics, interactions with the microenvironment being really, really important here as well. And so, as we've talked about a few times, I want to define my terms before we continue. When I say cell state, and kind of when our group discusses cell state, you can obviously define this in many different ways. We think about it as the group of genes that are co-expressed in individual cells that tell us something about the cell's function as well as the environment that it's sitting in right now. All right, so again, it's just as an overview for the group before we kind of dive into a couple of these topics and how they lead to things that bother me. We think a lot about which cell states matter and in what context, right? There's a lot of variability that we've detected over the course of the past, 10 years of sequencing single cells. How do we know which ones are actually useful and functional? We think about how do we link these to functional and targetable attributes. So we're actually, the downstream effect of all of our work at some point wants to be thinking about how do we aim drugs at the variation that we see in cell state, much like we see the targeted therapies work for cancer populations, right? Where you got a BRAF mutation, you can target the BRAF inhibitor. So we are largely spread across these disciplines currently, where we think a lot about how do we benchmark and engineer cellular models to better represent the patient features that are important. We think about mapping and understanding the context-specific gene functions, much kind of like what Tom Norman was presented on earlier today, but here using the much maligned ORFs instead of CRISPR-A, which we'll get into in a minute. And then with our partners at Microsoft, we think about how do we infer state behavior actually, and what types of data do we need to actually train models in a biologically useful way, and that is true to the underlying human phenotypes. Okay, so kind of one of the central questions that we think about in the group is, can these new modeling strategies actually help us to interpret these very high dimensional signals, and can they actually end up being better than a biologist at this interpretation? So we think about this from the data side, and we think about, do we actually have the right data sets to actually train any of these models in a useful way? So many of these biological foundation models, and many of you are better experts than I in this, they're really built on useful references. And actually, as Tom also alluded to earlier, they're only as good as the data sets that we put into them, right? So for things like the structure of the genome, for protein structure, we have pretty well annotated and agreed upon reference data sets. I think the other thing that's interesting about these measurements of biology is that they're much closer to first principle measurements than the next layer that we'll get to and that we've been discussing, right? An angstrom is an angstrom no matter who you talk to about what an angstrom is, right? That's not, we've had several discussions today about should I normalize, should I not normalize? That's a core piece of how you then interpret the following, RNA expression data. So that's not even that well agreed upon yet. So do we have these same things for RNA expression or cell state? So as we move up this kind of complexity chain, do we have these same types of reference data sets? Well, obviously, the elephant in the room is, we've got a ton of them. We've got single cell atlases. Like, what's the problem here? So one thing that has always bothered me when I was a postdoc was I felt like it was a race to the biggest. Like, who could give me the biggest atlas? Who could show me, 50 trillion cells versus, 40 trillion cells, and like that was going to be the next Nature paper. And so one thing that Lauren and I have argued about a lot, and not even really argued, we kind of both agree on this, is do I need to see that many cells to actually do these very basic classification techniques or classification problems that we use as a lot of benchmarks for these approaches? So obviously these are, again, I'm also not picking on any on any different foundation models here. These are just ones that were useful to show. We also benchmark against a couple of these. They're obviously trained in very large data sets, right? These are 30 million cells, and some of the most recent ones are even larger than that. The parameters are into the billions, right, of the models that end up being trained here. So this is a question that I would always ask either my immunologist friends or my computational friends, is how many T cells do I actually need to see to know that that's a T cell if I saw a brand new one, right? And so we had a really talented bunch of students, Alan, Madeline, and Akshaya kind of lead this effort to effectively do this experiment and figure out how many of these cells do I actually need to see to actually be able to do very simple classification tasks, okay? So we came up with a strategy to actually do this, where we set up a strategy to downsample a pretty large data set, a 22 million 22 million cell data corpus, where we then would either randomly subsample the data, we would cell type reweight sample the data, or perform geometric sketching just on the embedding, right? So we came up with all these different ways to do it. We then downsampled all the way from 100% of the data down to 1% of the data, and then we trained different model architectures. We then came up with several set of tasks for these models to do. So there was very simple things like cell type classification and relatively simple things like batch integration. And then for the fine-tuned tasks, we came up with cell type classification as well as perturbation prediction. At the end of the day, we trained, and this was one of the nice parts about being associated with Microsoft. I could never have done any of this, right? The amount of compute we kind of blew through just to test the even simple matrix that we came up with here, right? This is by no means exhaustive, but this was as exhaustive as we could be with the kind of the resources that we had. And I'm not going to go through a lot of the data here, but I just kind of want to point out that what we found was that regardless of model architecture, the performance peaks at a very small fraction of the data for these relatively simple tasks, right? So at as little as 1% of the input, we were fine at doing any of the tasks that we exposed these models to. So this might be true for the static representations of cells, right? But as we all know, cells are not just static, right? How do we know what they're doing in a dynamic fashion, right? So, okay, great. We've got single cell atlases that are at least help us get some part of the way there. But looking at the distribution of expression states, in clusters and pulling out marker genes, doesn't really tell us anything about the function, right? So what's really nice about a lot of these reference data sets is we know that, this sequence of amino acids will fold and at some level lead to this function, right, as a protein. We don't really have a map for how do groups of genes perform context-specific functions, right? If I've got a gene expression program that I find in a UMAP, should I care about it, or how do I label it? What do I do? What do I do about it? So this, again, by no means an exhaustive approach, but we've come up with a strategy where we're attempting to link kind of the variation that you can see, at least in model systems, to some type of functional output. So we call this a state to function map, where this is again led by a large group of scientists within the laboratory or within our lab, largely spearheaded by a very talented graduate student, now kind of a postdoc. She's on her way out, Evelyn Tong, who's going to be looking for a postdoctoral position. So I might be sending you all emails at some point. She's very, very talented. But anyway, so we come up with this, what they call at Microsoft our hypercube, where the idea is, can we actually engineer heterogeneity? Does this pointer work very well? No, use this. We can engineer heterogeneity. Again, you could do it the way that Tom has actually elegantly shown with CRISPR-A, which gives you this nice, actually physiologically relevant expression distribution. Or you can do it in the brute force way that we're doing it, which is with ORFs, right? Where I just kind of want to know if I turn on a transcription factor in a cell that either does or doesn't want it, can I elicit a phenotype, right? So we then expose each of these models to different environments, right? So we've got drug pressures that we'll put on them. We will put on things like inflammatory cytokines, nutrient deprivation, hypoxia. And we can start to learn which gene expression programs can control fitness under different pressures, right? So we've been using Again, Tom gave a great introduction to all of this, the Morph library that was built by Jessica Zhang when she was a postdoc with Feng Zhang in 2023, and it's covering all 1,800 TF transcription factor genes, and at the level of each isoform, actually. So we can start to get isoform level information from these screens, which is really interesting. I will say there's one technical problem with this approach, which is all of these ORFs are different lengths. So we absolutely get like a size bias, and we introduce this into our systems. And we're actually currently building a complementary set, very much like Tom's, to do a actually comparison of these different phenotypes. Please do.
I'm a little bit confused because you're talking about groups of genes,
Mm-hmm.
but those are all individual gene perturbations, right?
Right. Yep. So
How are you thinking about that?
yeah, great question. What we think about them as the transcription factors are just meant as probes to turn on gene expression. So when we then single cell sequence them,
CF gene.
exactly. Exactly. So what are the genes within, let's put it this way. I didn't have enough money to do the entire orpheome. That's also I think a little bit of a fool's errand if I'm looking at transcription factor. I'm sorry, if I'm looking at expression as the readout. You then couple this to single cell RNA sequencing downstream, and all of a sudden you've got modules of genes that are being controlled by that transcription factor in the different tissues. So yes, great question. Cool. So then we perform, the first layer that we do is a functional screen, right? So we run a fitness screen in this case. So here I'm showing one slice through one model, basically. I'm literally being pulled in, but it feels like it's too far away to gesture at, but then come all the way back here. Sorry, I like to be mobile. So we take one model and we can then show you one example here. This is a KRAS inhibitor in a KRAS mutant line. Please, yeah.
What the dose you selected for 7977?
Sorry, what?
For the KRAS inhibitor?
Yeah.
Which doses, like?
Oh, which doses? That's actually cell line specific. So we choose one that is going to, and actually you can kind of see it in this image. You choose one that's about 80%, like will kill the cells to about 80 to 90%, like of the starting population. So we
Collaborating with us, or you purchase this of the black market?
the black market. This drug you can just get this. You can get this drug.
It's like, are you collaborating with us, or you get it like from another company or like something?
Who? Oh, yeah. No, this is with Andy Aguirre. Yeah. Black market. Yeah. So we use this drug then as a probe for obviously a KRAS dependent line. So we use the doses that we use. It's actually a great question too. The doses that we end up using are cell line specific, right? So you want to choose a dose that will actually kill the cell well enough to actually see this signal. So what we're looking for is exactly what the bracket there suggests. We screen it against GFP cells and against the TF-ORF library to show that these are actually TF-induced changes, right?
Peter, is this multiple independent screens that you then assemble into each plane in your diagram?
Yeah, exactly.
Okay.
Yeah. So I'll show you some of that data in a minute, right? So that we can run individual screens. We usually try and keep them at the same time points, but it ends up being actually like a doubling cutoff is what we usually end up choosing for like the time scale. But it's almost always the same. We try and keep it because we know these things can drift in culture. We usually same split of the cell line, that type of thing. Yeah. So basically, then we can sequence the barcodes to figure out which TF which ORFs survived better in this condition. So that's for one. I'm going to cartoon the results here before I show you the real ones, where we can now actually, since this library is small, we can actually run it as a scalable screening strategy across many different parameters, right? So here we have in the KRAS mutant lines, we can actually see the TFs that are more fit in KRAS inhibitors, but now we can actually add things like inflammatory cytokines as well. And so then what I've always wanted to see from these data are, the hypothesis was there'd be shared set of transcription factors that it didn't matter which perturbation you threw at them, they would actually be fit in either condition. And so what's really interesting is that's what we end up seeing for, this is just one example screen. You're not really supposed to be able to read this. You'll see a little bit more of a summary on the next slide. But what's very cool is there's absolutely gene expression programs that can control fitness in one insult or the other. There's also ones that lie on this Y equals X line, right, where there are definitely convergent, let's say, functional programs that it doesn't matter, at least for these two. We've done several others that kind of tell us that some of these that are in the green here are truly convergent phenotypes. They will kind of handle a stress no matter what you throw at them. So this is something we've been very interested in. Now that's one that's one layer basically for one model. Oh yes, and to the point, we can then couple this to single cell RNA sequencing. You can get all the programs. It's tons of data. It's been really annoying to sift through actually. But what's very cool then is you can now go through different tissue types using the exact same perturbations, and we can build these maps in a much more comprehensive way. Here is an example of just for KRAS inhibitors and for inflammatory cytokines, where there are now, we've done this across 8+ models across 4 different tissue types, where you can see there's absolutely recurrent features of fitness in these different contexts. Okay. So great. What if we keep doing this, right? So now we've got one way to map functions. There's many other ways. There's people doing very elegant studies to actually learn this as well. But I'm kind of going to undercut everything I just said by posing the question, once we've done this, we're going to be fine, right? We'll have learned how a cell functions, and we can train a giant model and have a virtual cell.
Well, I mean,
Yeah.
your collaborator might tell you already that the things that were in that shared access, some of them are not that relevant sometimes in patients, not so.
Exactly. So that's why I breezed past it, but you either compare the TF levels in patients or you compare the gene expression programs in patients, and this allows us to sift basically ones that this is a quirk of tissue culture biology that's cool maybe to understand from a systems biology standpoint, but for patients it doesn't really matter. So I could show you another plot where you overlay patient relevant or not patient relevant, right? So you're exactly right. So our North Star is, do I see it in patient data? And that's what either happens or doesn't happen for a lot of these. So it's a great segue though. The question is then, is classic systems biology actually human systems biology, right? So we can get really good at this circle of iterative perturbing, measuring, and analyzing within more simple, more perturbable systems like yeast, zebrafish. We can learn that really well, right? Because there is no disjointed connection between the two. But that's not true for human biology, right? So we do all this discovery in patient cohorts. We then literally have to incur what we've kind of coined as this idea of translational distance between the human and whatever model system you're using. So here, do we actually account for those assumptions between our human and model systems? So there's this big problem for all of our model systems that we actually have right now. It's one that I think everyone acknowledges, but we don't really do a great job of triaging. So this is one example of, in brain tumors, none of these models actually ever aligned with the patient tumors they were supposed to come from. So here you can see in blue is the 2D cell lines that we use from DepMap, and in this peachy color are the actual tumors they're supposed to align with. And there's literally no overlap between these two populations. So many of these 2D and 3D models actually don't represent the phenotypes that we think they do. And this largely stems from the idea that a model was considered successful if it grew and if the genetics were correct. But we never really checked all of the downstream expression states, were those correct, that type of thing. The conditions are really not tuned in many of these aspects for capturing the expression states that we're interested in. Moreover, are they phenotypically and functionally aligned? So they're also, unfortunately, not functionally aligned in a lot of cases. So here, if you took existing cell lines, this is from actually a really nice paper from 2011, where this axis of heterogeneity was discovered. In pancreas cancer, there's principally two states that matter, the basal and the classical state. This early paper said that, hey, the non-responders to chemotherapy are going to be in this classical state. What's been interesting is that now, as the clinical trial data have come out, the classical, that's actually totally incorrect, right? So the classical tumors are actually the ones that respond pretty well to chemotherapy, and the basal tumors actually resist it, right? So the problem being that many of our existing cell lines just don't mirror the functional patient attributes at all, but we've actually shown that the functional alignment can be possible at some level. So the question then becomes, do we actually properly consider what we are learning with all this perturbational data that we're making from our cell line models, from our organoid models, things like that? All right, so I'll finish on this slide of this idea that has been vexing me over the past couple of years, this idea of the perturbation and fidelity trade-off, right? Where we build all these very dense perturbational data sets in things like cell lines, where we can do a lot of really elegant, deep, comprehensive scanning, right? But we're about as far away from the human condition as you could be. There's again beautiful work that has been done over the past several years, actually by many people in this room. Peter Sims actually showed a great platform the other day to get as close to human biology as possible and start to perturb. But the problem becomes could we ever get to a comprehensive map of how a high fidelity human model behaves when you perturb it with about 10 at a time, right? I just don't know if we're going to be able to get there. So the place that I've been interested in thinking about is, how do we get to this kind of unoccupied frontier? And I think what's been cool over the last several years is there are some that start to kind of inch into this area where we take primary cultures and we start to do really large scale perturbation screens in them. But I kind of want to finish on this idea of what's it going to take to get there? Right. My suggestion is I think that's where we need to be. I don't know that a model can ever learn, if I do all this great perturbation over here in cell lines, can it actually ever learn the human condition if it's never seen it, right? So is it going to be an AI? Is it going to be a better trained model? Is it going to be new experimental techniques? I think like these purple arrows are the thing that kind of keeps me up at night personally. But yeah, so with that, I mean, I'd be happy to take any questions or we could discuss later on.
Yeah, so I think that we're running a little, have to take questions offline. But thank you so much for the next.